Hello, Bio 111. This is Chapter 3, Biological Macromolecules. I'm going to go over the entire chapter in this video. Again, feel free to pause whenever you'd like. We will reach some points where there's some activities, and I will give you the chance to pause and figure it out before I show you the answer. Of course, take notes where you feel necessary, and let's get started. Okay, so 3.1, section 3.1, the objectives, we're going to understand macromolecule synthesis and explain dehydration and hydrolysis reactions. Those terms might seem kind of large and crazy to you, maybe you don't understand them, but hopefully by the end you understand the terms and what they mean. So here we go. So there are four major classes of biological macromolecules. Sometimes they might be referred to just as macromolecules. Sometimes you might hear them as biomacromolecules. So there's carbohydrates, lipids, proteins, and nucleic acids. And over here on the right, there's a nice graphic showing you the breakdown or what that macromolecule might look like on a element and molecule scale, very small. So these are organic molecules. So if you ever take organic chemistry, you are going to look at mainly these four molecules. And they're organic because they contain carbon. So if you take a closer look at these molecules, you can see there's a lot of C's in these, right? A lot of C's. There are also C's at the points where they're not labeled, and those are carbon. These molecules also contain hydrogen, oxygen, nitrogen, and some other minor elements. But carbon is great because you can make four stable bonds with carbon. So if we take a closer look at this lipid, this carbon here can make four bonds, and that's mainly why everything is made up of carbon. Now, there's silicon as well, which can make four stable bonds, but it's a heavier element, and we have more carbon on Earth than silicon. So I think that's why all organic life is based off of carbon. So it's based off of these four molecules. So that's what we're going to learn about. You also might understand that these four things are part of what you eat every day. Carbohydrates sounds very familiar. Lipids is a fat, proteins, and we don't really get into nucleic acids when we talk about food, but these are four things that your food contain. So, very important to you. Biological macromolecules consist of subunits called monomers. So monomers are like building blocks, and we can stack them together to make a larger unit, which is called a polymer. So a monomer is just one small molecule, but if we repeat that one small molecule over and over again, we get a polymer. And so monomer and polymer are just the basic terms. Most of the time, each of the four classes have their own special name for that polymer. So for instance, an amino acid or a nucleic acid is going to build on itself. So those are monomers and polymers. But we'll get into that a little bit more here in a bit. So not getting into specifics with that right now, but monomers and polymers, we can connect them in certain ways. So we're going to talk about the bonds that they form. So here's a breakdown of polymers and monomers. You can come back to this page at any time. You might want to screenshot this. This is a nice cheat sheet to see what the monomer and the polymer are called for each of the four classes that we talk about. Okay. All right, so we can make monomers, or we can combine monomers, rather, into polymers by going through a chemical reaction. So making a polymer, we're adding monomers together to make a polymer. It's called dehydration synthesis. Big term, right? So dehydration synthesis, two monomers added together make this small polymer. And it's called putting together while losing water. So you can see these red H and OH here, these two things are going to combine, and with the combination of those two things, now we can form a bond. So we remove H and OH, and now we have space to make a bond here. So this is how a polymer can be created. So we're going to lose water when we do that. Dehydration synthesis, right? So we're losing water, we're dehydrating those monomers, and we're synthesizing a larger molecule. Okay, does that make sense? So a water molecule formed by combining, and they are combined by a covalent bond. Now, if you want to go back to chapter 2, where it talks about bonds and covalent bonds and ionic bonds and all that, you can go back and read about chapter 2. We're not going to get into all the bonds, but covalent bond is a nice bond because it is very stable. Okay, so dehydration synthesis leads to the formation of what of the following? Take your time, you can pause. All right, so the answer is C, water, water and polymers. I combined water and polymers. So letter C, so we're gonna make a polymer, and then we're also making water. And of course, we're not making it out of nothing, right? So we need our polymers there to make that, we need those monomers there to make that polymer. Okay, so the opposite of dehydration synthesis is hydrolysis. So it's the opposite, reverse reaction. We're breaking down that large molecule into smaller molecules, which usually are easier digested or easy to work with, right? A big unit, a big polymer is not easy for your body to work with. Your body would rather break it down. So this is digestion. So big polymer, right? And polymers can be two subunits long. They can be 30 subunits long. They could be over 100 subunits long. This is just a simplified one to show you the basics. So this polymer, by adding water, so by drinking water and eating the polymer, your body can break it down, right, by adding that water back to it. And so now we have two monomers, two subunits that are separated from each other, and now your body can handle it a little bit better. We can utilize those monomers by doing other things with them, getting energy from them. Okay. So another question, you can pause here if you'd like. During the breakdown of polymers, which of the following reactions take place? So we're breaking down polymers, which should be letter A. So hydrolysis is the correct answer. Good job if you got that right. Now there is a really nice video for you to watch on your own time. There's the link there. There's also an interactive about the two reactions at the bottom. So if you want to copy that URL down, you can if you want some extra practice. But I'm going to ask you one more question before we move on. All right, so you're looking at 3 different reactions, 1, 2, and 3. What I want you to determine is which of those is dehydration and which of those is hydrolysis. Of course, you're going to use one of them twice. So go ahead and pause it, take your time to decide. Okay, so number 1 is dehydration synthesis. Because we are making a polymer, we're synthesizing a polymer and losing water. Number 2 is hydrolysis. We are taking a big polymer with water and breaking it down into 2 smaller parts. And then the last one is dehydration synthesis. So 2 monomers making that polymer, and we're losing water. And the reverse reaction would be hydrolysis. So that one was kind of a which one are you talking about situation there. So extension, what purpose does drinking water serve? Well, it's digestion, right? We need water to break down those giant polymers. If we're dehydrated, then we can't do digestion properly. So that's why we need to drink water on a regular basis. Okay, so these reactions can happen at their own rate, which is okay, but they can also be sped up by using an enzyme. So enzymes are biological molecules, they're a protein, that catalyze or speed up reactions. So they speed up chemical reactions. Enzymes can be used for either hydrolysis or dehydration synthesis reactions. They are very specific on what they do. So an enzyme that breaks down sucrose is only going to break down sucrose. And then an enzyme that would build sucrose would only build sucrose. Okay, so it could only do those certain things. And there are a lot of enzymes for each macromolecule class. There are also enzymes for nucleic acids, for building nucleic acids as well. I just didn't list them here. But these are the common ones that you will see. I did underline the ASE on purpose because most of the enzymes that you see end in ASE. So if you see a word that ends in that, it is typically going to be an enzyme. Okay, so some of the enzymes in your body can break down sugars, right? But you also are lacking some enzymes that break down artificial sugar. So that's why your body can't really digest artificial sugar. So things like Splenda. So the sucralose that makes up Splenda, your body can't break it down, which means it can't get the energy from it. So it's no calories. It's a no-calorie sweetener because your body doesn't have the enzymes to use it. So it just kind of passes throughout your body. Some of it is stored because it's actually a toxin. But anyway, let's go on to the next section. So hopefully now you understand what macromolecule synthesis is, and you understand dehydration and hydrolysis reactions. Okay, next section. So we're going to talk about each of those 4 subclasses of biological macromolecules. So first we're going to cover carbohydrates. So by the end of this section, you should understand what the role of carbohydrates is in cells. And on the outside of cells, we'll explain some carbohydrate classifications. And so here, there's some classifications down below: monosaccharides, disaccharides, polysaccharides. Hopefully you can list a couple of each of those by the end of this section. Okay, carbohydrates. Typically when you hear the word carbohydrate, you think sugar, you think bread, pasta, corn, fruit, potatoes, right? Those are things that contain large amounts of carbohydrates. So they provide our energy in the form, our body energy in the form of glucose. So carbohydrates are mostly made up of glucose or some form like glucose. And so they are really large polymers that your body has to digest, break down, do hydrolysis, right? And then we can use the monomer subunits. Okay, so typically you're going to see carbohydrates represented in some form like this. So CH2O to the N multiplication factor. So C6H12O6 is typical glucose. So that is just one monomer of a carbohydrate, right? So that's, we're going to have twice as much hydrogen than carbon and oxygen is basically what it's saying. And same with that ratio down there, carbon to hydrogen to oxygen. So 3 main subtypes are monosaccharides, disaccharides, and polysaccharides. Okay, so monosaccharide is a monomer. Disaccharide is 2 monomers together, and then a polysaccharide is more than 2. All right, so monosaccharides, monomers of carbohydrates, typically 1 ring. They usually have 3 to 7 carbons. So remember, all of these 4 divisions of macromolecules are going to be carbon-based. So we're very interested in carbon here in this chapter. They have one ring, 3 to 7 carbons. They usually end in the suffix OSE. Now that's not all the time, but it's most of the time. So glucose, common one, right? Fructose, and then galactose. So these are the typical monosaccharides that we're going to see in the real world. Okay, so I know probably blowing your mind with this page. You're like, whoa, it's too much. So here we go. Structural isomers formula. So an isomer, it has the same chemical formula, but it's just arranged a little differently. So glucose, galactose, and fructose here, they all are C6H12O6, but they're all arranged a little differently. So you can see this end one is pretty obvious. That double bond to oxygen is in a different spot. And then glucose and galactose, the middle parts here are arranged a little differently. So these are different enough to call them different monosaccharides, right? And they come from different places. So glucose is what plants synthesize during photosynthesis, okay? Galactose is part of lactose, so milk. So animals make galactose. We make galactose. Fructose is part of fruits, so the storage portion of plants is where fructose comes in. It's very sweet. And so these are, again, 3 monosaccharides. So hopefully you can name these 3 when I ask what is a monosaccharide. You can say, oh, well, it's glucose, it's galactose, it's fructose. A monosaccharide is just one subunit, right? It's a monomer. So we can add these things together to make polymers, okay? Okay, so normally if you had a model making kit, I would have you create one of these on your own, but you don't, so it's okay. We can go on to the next slide. So monosaccharides are what? Glucose, galactose, fructose. So disaccharides are combining any combination of those monosaccharides together. So remember, glucose is made by plants. Fructose is made in the fruit part of the plant. If we put those 2 things together, we get sucrose. So that is a disaccharide. There are 2 monomer subunits. Sucrose is a table sugar. So the sugar that you buy at the grocery store that comes in that paper bag, that's sucrose. So it's a disaccharide, which is pretty neat. They are combined together through what's called a glycosidic bond. It's just a nice term for covalent bond. It's a specific term for a disaccharide covalent bond. Okay, so sucrose. Let's look at some other disaccharides. Maltose and lactose are other disaccharides. So maltose is grain sugar. So that's 2 glucoses added together. So grain sugar is like wheat, barley, rice. Those types of things make maltose, and that's their storage carbohydrate, right? So when you eat rice or you eat oatmeal or bread, you're consuming a lot of maltose. Lactose, again, so that's what animals make. That's what we make. Milk sugar is what it's called. And that is a glucose and a galactose together. Okay, so 2 of those monosaccharides added together. And then we already covered sucrose, which is glucose, and fructose added together. Okay, so again, disaccharides are just 2 monomers, 2 monosaccharides added together. So a polysaccharide is a large carbohydrate. It has more than 2 subunits, and it can be extremely large. So their molecular weight could be over 10,000 AMU, which is huge. Glycogen is one polysaccharide that's in our bodies. We use it as a storage function. Animals use it as a storage carbohydrate. So it's stable. When we run out of food or we're not eating enough food, our body will break down glycogen. So it's nice. It's a nice storage function. It also insulates our insides. So polysaccharides can be huge, they can be branched, they can be straight, they can be they can consist of multiple types of monosaccharides. So we looked at the disaccharides and they had different combinations. Polysaccharides can also have different combinations. So this is what I mean by straight or branched. Amylose is a starch. And so is amylopectin, but this one's branched here, so that one's straight. So they're just different on how they're arranged. These are sources of these starches, so they tend to be stored in plants, right? Okay. Cellulose is another polysaccharide made by plants, but this one is placed in the cell wall. So remember, plant cells, they have a cell membrane like normal, and then they also have a cell wall, which is on the exterior of that cell membrane. And its cell wall is just mostly for structure. Remember, plants don't have bones, so they have this tough cell wall, which is made of cellulose, and that is a polysaccharide. Okay, so it's nice and straight, and it's just this repeated over and over for forever, right? So nice fibrous structure, not easily broken down by animals. Chitin is another polysaccharide, and this is in the exoskeleton of arthropods. It's also in the cell wall of fungi. So fungi, remember, they're not plants. They're their own thing, and so they have different structures. So their cell wall, or their cell wall-like structure, is made of chitin. And chitin's pretty neat. It's made of nitrogen, which is different than most polysaccharides. Okay, so back to glycogen. Glycogen is what we have. This here is one molecule of glycogen. So each of these red little structures is a monosaccharide. Just take a second and look at how many there are. Crazy how big it is. But it's a really nice structure. And then there's a, so each of the red is the carbohydrate portion. This middle portion is a protein, which helps keep it in place. So each of these red structures again, so it's a monosaccharide, and that's how we store extra carbohydrates. So when we overeat and we eat too many carbohydrates, our body starts to store them as glycogen. So your body usually stores glycogen in your liver. It also stores it in adipose tissue, which is fat tissue. It could store it in your blood, and it can store it in your muscles as well. So storing it in your blood is probably the worst place for it, especially if you're a diabetic. But that's where glycogen is stored. And it is made to be broken down. So hopefully your body will break it down in the future. There's a nice video here for carbohydrates review if you want to go back over that. But let's go on to the poll question. Which of the following is not an extracellular matrix role of carbohydrates? So extracellular matrix is just referring to that outside covering of the cell. So that was like chitin in the arthropod, that was like the cell wall in the plant, cell wall in the fungi, storage in those plant structures. So the correct answer is A, protect an insect's internal organs from external trauma. So that's not a role of carbohydrates for insects, but in animals, if we were talking about protect an animal's internal organs, yeah, that would be the correct answer. So that would make that answer correct. All right, so that's the end of carbohydrates. Hopefully now you can discuss the role of carbohydrates, explain some classifications, and then list some common monosaccharide, disaccharide, and polysaccharides. So next up we have lipids. So we're going to describe the 4 major types of lipids. We will explain the role of fats, and we will differentiate between saturated and unsaturated fatty acids. So you probably saw these listed on a nutrition label before, so we're going to talk about those. We will discuss phospholipids, and we will describe what a steroid is and what cholesterol is. Okay, so lipids are a diverse group of non-polar hydrocarbons. So non-polar means that the molecule is hydrophobic. So that means it does not like water. So hopefully this meme over here will help you understand that. So here is a lipid, here's water. They can't be together because the lipid hates water. It repels water. And they're a hydrocarbon because there are a lot of carbons and a lot of hydrogens added together to make this molecule. Okay, so sometimes you might hear a hydrocarbon as a term when somebody talks about gasoline. And so that's kind of the same thing, but let's talk about lipids here in a sec. Or let's get back to lipids. So lipids, functions of lipids, long-term energy stores. They provide lasting energy, so it takes your body a little longer to break down lipids. Your body breaks down carbohydrates pretty quickly, but it takes it a little bit longer to break down lipids. Lipids also help insulate animals from the environment. So this otter here has a lot of lipids on its fur. So it has like a waxy coating on its fur, but it also has a layer of fat underneath of its skin that helps insulate it. Lipids also serve as the building blocks for some hormones, and they are an important component of cell membranes. Okay, so the 4 classes of lipids, so remember this was one of our objectives. 4 classes are fats and oils, waxes, phospholipids, and steroids. Okay, so we're going to talk about each of these here. We're going to break them down for you, tell you what they are. So here we go. All right, fats and oils. So fats contain 2 components. This is a fat molecule here. Okay, fats and oils. Remember, fats and oils. Not all lipids are fats, right? So this is just a fat, which is part of a lipid, which is one classification of a lipid. So they are made up of a glycerol, which is here, and then they are made up of fatty acid tails, which run that way. So this is called a triglyceride, and it is a triglyceride because there are 3 fatty acid tails and that glycerol head. So this glycerol is actually technically an alcohol because it has a lot of those OHs. It ends in the OL. That's how you know it's an alcohol. Easy to remember that one, right? So a triglyceride is an alcohol, glycerol, and then 3 of those fatty acid tails. So these fatty acid tails can be different lengths. They can be different structures, which I'll show you in a second. So they might not always look like this. So just keep that in mind when we get to the next few slides. Okay, so this is a fatty acid tail. This is a saturated fatty acid tail. So when I say saturated, it means that it's saturated with hydrogens. Or you could even say saturated with single bonds. So all of these carbons have single bonds to hydrogen. There are hydrogens on each side of those carbohydrates. So they are very straight. They can be packed very tightly. And typically, when we're talking about food, saturated fatty acids are solid at room temperature because they can pack in so tightly. So that's butter, that's lard or fats in meats, things like coconut oil, cheese, even cream. Those are things that are solid at room temperature. So these things are also associated with cardiovascular disease. Your doctor has probably told you, or somebody has told you, to limit saturated fatty acids in your diet because they are not that great for you. Of course, a little bit is okay, but not a lot. Unsaturated fatty acids are better than saturated because they have this bend to them. So usually they contain at least one double bond. So when I say an unsaturated fatty acid, I mean they are unsaturated with hydrogen. So look at, there's two that are missing right here. So they're unsaturated. You can have monounsaturated fats, which is just one double bond, or you can have polyunsaturated fats, which is more than one double bond. Most unsaturated fats are liquids at room temperatures. So those are oils. So monounsaturated fats are like olive oil, canola oil, peanut oil, sesame oil, avocados, or avocado oil, and most nuts have monounsaturated fats in them. Polyunsaturated fats are things like corn oil, sunflower oil, soybean oil seeds, cold water fish. So those are the better fats to consume for your health. So what's the deal with trans fats? I'm sure you've heard trans fats are not good for you. They do have a double bond, but that double bond does not make it curve like it usually does. So this is it's called a cis unsaturated fatty acid, and this is a trans unsaturated fatty acid. So that cis is just referring to that the hydrogens are on the same side, and then the trans is referring to the hydrogens are on opposite sides of each other. So cis fatty acids are good. They have a kink in the chain. Typically they're liquids at room temperature. Trans can be straight, typically hydrogenated oil. You've probably heard that term before. This is how trans fats are made, through processing. So hydrogenated oil is not good. That's a trans fat. Foods with trans fat may increase LDL cholesterol, which is not a good thing either. Okay, so cis unsaturated fatty acids are great, but double check that nutrition label to see if there are also trans fats, and if there are trans fats, it's not good. So if you have a modeling kit, you can practice creating one. If not, we can move right along. All right, lipids. So fatty acid, these are essential fatty acids. Lipids that are shaped in this hook-like pattern, which is kind of funny to me, are things that are from fish. Hook-like pattern, they're from fish. It's kind of funny. So these are called omega fats. There's omega-3 and omega-6 fatty acids. So salmon, trout, tuna are sources of omega-3, which is good for a healthy heart. We cannot synthesize omega-3s ourselves, so we have to get them from our diet. So that's why eating fish once a week or a couple of times a week is a good thing to do. Omega-6 fatty acids are found in oils, so if you cook your fish in some oil, then you're doing double duty. All right, so poll question. Saturated fats all have what? Except. Saturated fats have all the following characteristics except what? That is letter D. They tend to dissolve in water easily. So remember our first couple slides about lipids, they are hydrophobic. So they do not like water, which means that they are not going to dissolve in water either. All right, going on. So waxes, I only have one slide for waxes because they are so different depending on which wax you're talking about. So waxes, they can have chains, they can have rings on them. They are hydrophobic, just like the rest of lipids. They don't like water. They prevent water from sticking to the surface. Typically found on aquatic animals, aquatic birds, and plants. So like this holly bush here, very waxy. Helps protect it. All right, phospholipids. So phospholipids are found in the cell membrane. So all the cells in your body have a cell membrane, and phospholipids make up that membrane. So phospholipids are different. They are I say almost, like a triglyceride. So they have that glycerol backbone, but instead of that third fatty acid tail, they have this part instead, which is a phosphate group. So that makes them have a few different chemical characteristics. So phospholipids typically look like this in a molecular scale, and we might refer to them like that when we draw a cell membrane. So they have this head region, which is where the glycerol and the phosphate are, and then they have the tail region. So the head region is going to be hydrophilic. So remember, lipids hate water. Okay, lipids hate water. But phospholipids can tolerate water because of that phosphate group, which is located in the head region. So the head region can face towards the liquidy water portion of the cell, and then the outside portion of the cell, which may contain water, which is great. And then the inside portion where the fatty acid tails are, they still hate water, so they're going to be protected in that sandwich layer. I like this meme over here. It says, hey guys, look, I have a water gun. And they're all like, no, moving away from the water, so they don't like it. Okay. Steroids. Steroids are the last type of lipid that we'll cover. Steroids have these rings to them. They still, they're like other lipids. They don't like water. Many of them will have this short tail to them. Sometimes they might not have a tail at all. This one here is cholesterol, and this one is cortisol. So cholesterol is a steroid. Bet you didn't know that, so learn something new. So again, they are hydrophobic. They do not dissolve in water. Cholesterol is the most common steroid in your body. It is made by the liver. It is a precursor to other hormones. So testosterone, estradiol, those are hormones, right? And they're made up of steroids. Precursor to vitamin D, and then precursor to bile salts as well. So steroids or cholesterol is not all bad. Of course, too much of a good thing is bad, right? But we still need it. So steroids are not a carbohydrate, right? They look like a carbohydrate a little bit because they have that ring structure, but they're not a carbohydrate because they are hydrophobic. They don't like water. So phospholipids are an important component of what? Letter A, the plasma membrane of cells. Okay, so that brings us to the end of lipids. So hopefully now you can describe the four types. So remember the four types are what? Fats and oils, phospholipids, waxes, and steroids. We store glycerol, or not glycerol, glycogen in our body for later use. That's our way of storing fat. We can also store fat to help insulate our body. We can use fat as an insulation and a waterproofing. Saturated fats are those straight chains. They are solids at room temperature. Unsaturated, cis unsaturated fat are the bent ones. Those are liquids. Trans unsaturated fat are the straight ones with double bonds. Those are bad. Those are solids as well. Describe phospholipids. So phospholipids make up the cell membrane. We will talk more about the cell membrane when we get to chapter 5. The basic structure of a steroid is a ring structure, usually has 4 rings. Cholesterol is a steroid, also makes hormones, and it helps make the plasma membrane fluid, which we may not have talked about, but it does. It helps make it fluid because it helps provide some spacing in the membrane. All right, so 3.4 proteins. We're going to describe functions of proteins. We'll talk about the relationship of amino acids and proteins. We'll talk about the 4 levels of organization, which is what this GIF is showing you. And then we will talk about the structure and shape of proteins and why that's important. So primary, secondary, tertiary, quaternary is the 4 levels of protein organization. So we'll talk about that there in a second. Proteins are the most abundant organic molecules, so we have a lot of protein in our body. We have a lot of different protein in our body. So proteins have a very diverse range of functions, but each protein is very specific on what it does. So you can have regulatory function protein, structural function protein, proteins that provide protection, proteins that transport molecules, proteins that act as enzymes. And we already talked about enzymes, right? So we know how specific enzymes are. And then proteins that are even toxins for your body. So here is a list of protein types and functions. This is straight out of your textbook. So you can see here's some digestive enzymes that we've talked about before. So that helps break down food, and it also could help build other enzymes. Let's see, storage enzymes or storage proteins provide nourishment of like an embryo or seedling, defense and all that. So enzymes again. So I told you we'd come back to enzymes. Enzymes are a special protein. They help speed up reactions. So the reaction could be that it's building something or it's breaking something down. So if we're talking about an enzyme in your stomach that is breaking things down, that would be a catabolic enzyme. Catabolic enzyme, okay? If we're talking about an enzyme elsewhere in your body that's building protein, so let's say like in a muscle, that's going to build protein, that's an anabolic enzyme. So anabolic, I remember that one because it has an A, so it's like adding things together. So adding things, building things, that's an anabolic enzyme, okay? Catabolic is the opposite. So this is what an enzyme would look like on a smaller scale. Of course, each of these would be little molecules in here. This is just a simplified version, even though it looks very complex. This is an enzyme that catalyzes the conversion of maltose to glucose. So we're breaking down maltose. It has it's a disaccharide, right? We're breaking it down into its subparts, which is glucose, 2-glucose. So this curly white shaded object here is the enzyme, the protein itself. And then this little space here is where it's going to break down those parts. So enzymes, again, very specific. It only functions on one thing. So this enzyme is only ever going to break down maltose. It's not going to break down fructose or sucrose, right? Because they just won't fit. The substrate would be different. So it's only going to fit that one substrate. So proteins are made up of amino acids. That's the monomer. The protein is the polymer. So their fundamental structure is this right here. So this is an amino acid. That's just one. Now we're going to link these together, right? Link these together. So this is the separation by what? Dehydration synthesis, right? So this is this is 2 amino acids together. So each amino acid, and I say each because there's 20 different amino acids. Each amino acid has a carboxyl group, it has this amino group, it has an R side chain. So you might say like, oh, I don't know, I've never seen an R before when we're talking about molecules. This R is just a placeholder for the different amino acid R side chains. So again, there are 20 amino acids. Each one has a different R group. The R group can be different in the chemical nature that it has. So it could be nonpolar, it could be polar, which means it's going to repel or like water. It could be positively charged, it could be negatively charged, it could be a nonpolar ring, like our guy here, this is a ring. So let's take a look at each of them. Here's 20 common amino acids. So again, these, the things that you are looking at are the things shaded in blue. So those are each of the R groups. The things not shaded in blue is the rest of the amino acid that remains the same for each amino acid. Okay, so again, the only thing changing on each of these is what's shaded in blue, and that determines its chemical nature. So we have glycine, alanine, valine. These are nonpolar. These are nonpolar R groups. And then down here, things like cysteine, serine, those are polar. And then lysine, arginine, histidine are positively charged. You can see the little plus signs here. Then there's negatively charged, which is aspartate and glutamate, negative, right? And then you have those ring structures, which are nonpolar. So each of these amino acids have different chemical makeups. They have different chemical formulas, and if we arrange them in certain ways, they're going to cause the overall function of the protein to be different. So if we have a lot of nonpolar amino acids linked together, it's not going to like water, right? So it's going to be in a place that doesn't like water. So maybe it'll situate itself in the plasma membrane. Okay, let's see. Which amino acid is this guy here? So this part is the COOH. That's our nitrogen group there. And this, where my red dot is the central carbon. So this bend and then this ring is the amino acid. So which amino acid is that right there? And if you said phenylalanine, then you'd be correct, right? Because it has that carbon, which is represented by that bend, and then it has that ring. So that's phenylalanine. So that's a nonpolar aromatic amino acid. And again, there are 20 of them, so that's just one of the 20. All right, so we can make polymers, we can make proteins by linking the amino acids together. So they're linked in what's called a peptide bond, which is a type of covalent bond. Again, this is dehydration synthesis, so water is going to be released, right? This H and OH are going to combine, they're going to be shuttled off somewhere, and then that leaves room for a bond to occur. So again, that's a peptide formation. So proteins, you might hear them called polypeptides, protein, polypeptide, typically interchangeable, and they're formed by peptide bond linkage. All right, so protein shape is based on 4 levels of structure: primary, secondary, tertiary, and quaternary. We're going to look at each of these. A protein shape is very critical to its function. So each of these structures, if something happens and that structure is not what it should be, then the protein's function is not going to be what it should be. It's not going to occur correctly. It's not going to happen. So the first one is primary structure. Primary structure is the unique sequence of amino acids. So it's which amino acid are we using, and then what order is that amino acid in with the other amino acids. So remember, we had a lot of different amino acids. There were polar, there were nonpolar charged, so on and so forth. So which amino acids we used and which order they're in is very specific. And it is based on your genes in your DNA. So your DNA is going to tell your body, like, hey, we're going to make insulin, right? So this is insulin, and it has to be in this order. All the amino acids have to be in this exact order, or it's not going to function correctly. It's going to be a dysfunctional insulin protein. So primary structure is very specific in every protein that we make. Another protein that's very common is hemoglobin. And hemoglobin can become dysfunctional and it can become sickle-shaped. And the sickle shape is only because of one amino acid substitution. So instead of this glutamic acid, there's a valine instead. So if you look, threonine, proline, glutamic acid, lysine, threonine, proline, valine, lysine. So just that one substitution causes this entire shape to be sickle-shaped. And so if you know what sickle cell anemia is, that's not a good thing to have. It makes breathing difficult. It makes a lot of health issues. And so it's just that one substitution in the primary structure that causes that to happen. It's crazy that it's only that one little thing that makes that problem. The next structure is secondary structure. So secondary structure creates either a pleated sheet or this curlicue helix. And that is just due to hydrogen bonding between the carbon, the COOH group, and the nitrogen group on each amino acid. So if you notice, here's our little R group, R group, R group. So these are each amino acids that we're looking at. So the interaction between two adjacent amino acids would create these structures. So again, helix and pleated sheet. Tertiary protein structure is more chemical interactions, but they are between R groups instead of COOH and N groups. So they could be things like ionic bonding, which is positive and negative attraction of R groups. They can be hydrophobic interactions, so those would be contained like on the inside of a protein, right? Hydrophobic, they don't like water, so they would probably like be protected a little bit in the center of a protein. If there are sulfur on the R groups, those are going to be linked in a disulfide linkage. And then hydrogen bonding can also occur still. So these are tertiary structures of proteins. Some proteins end at this point where they only have a tertiary structure. Most proteins have a quaternary structure. So a quaternary structure would be other tertiary structures added together. So this red, orange, blue, and green, those are separate units. So those are separate proteins. But if we add them together, we get a quaternary structure. And the weak interactions of those subunits help create the structure and help stabilize the structure. So the one that we're looking at there is actually hemoglobin. Okay. And then a red blood cell contains about 270 million of these protein molecules. Just one RBC contains 270 million of those. And right in the center of each of these is where oxygen will bind. So to sum up protein structure, these are the four, so primary, secondary, tertiary, and quaternary. Okay, so I said at the beginning where we went over the objectives for protein that structure and function go hand in hand. So if we denature the protein, we would change its function, and we might actually change it to where it can't change back. So chemical interactions are broken in the protein, which causes its function to change. So changes in pH, changes in temperature, and interactions with other chemicals, and maybe even like radiation, would cause denaturation of a protein. So if you think about an egg has a lot of protein in it. When we put it in a hot frying pan, it's going to change color, and that actually changes the protein shapes in the egg itself. If you take a protein from your stomach, like pepsin. Pepsin likes a very low pH, like a pH of 2. If you take pepsin and you put it in like a higher pH, like pH 7, it's not going to work anymore because it's outside of its normal range. So changes in pH from where the protein likes to be situated are going to cause deformation of the protein. All right, if you like review videos, here's another good one that I found online. Poll question to sum up proteins. So mad cow disease is an infectious disease where one misfolded protein causes all other copies of the protein to begin misfolding. This is an example of a disease impacting which structure? What do you think? That would be letter C. So tertiary structure. So one misfolded protein, one misfolded tertiary structure causes the rest of the structure to begin to be misfolded. All right, so now you should understand these ones, these objectives for proteins. Hopefully you can describe the function, the relationship between amino acids and proteins. So that's the monomer, that's the polymer. Explain the 4 levels: primary, secondary, tertiary, quaternary, and then the ways where we can denature. So changes in pH, temperature, interactions with chemicals, and even radiation. All right, last but not least, nucleic acids. This one will be short. We're going to describe nucleic acid structure. We're going to look at DNA and RNA. So later on in the course, towards the end, we talk more about DNA and RNA. So we're just going to cover some of the basics here. Nucleic acids constitute genetic material, genetic information of living organisms. All living organisms have nucleic acids. So living organisms rely on nucleic acids to record information and save information, and then they pass that information down to their offspring. 2 types of nucleic acids are deoxyribonucleic acid and ribonucleic acid. So DNA and RNA. The location of nucleic acids is going to be in one of 4 places or multiple places. The nucleus of eukaryotic cells, the mitochondria organelle, the chloroplast organelle, and then in prokaryotic cells, they don't have a nucleus, so it's just hanging out in there loose or connected to the cell membrane. But we'll talk about prokaryotic cells later on. All right, nucleic acids. So nucleotide, the nucleotide is the monomer. So the monomer of a nucleic acid, nucleotide, right? So a nucleotide has 3 parts. So an amino acid that we talked about in protein has a few parts in it, right? A nucleotide also has a few parts. So again, nucleotide is the monomer. It contains a nitrogenous base, which could be one of 5 things. It has a pentose sugar, and then it has a phosphate group. And it could have 1, it could have 2, it could have 3. This sugar could be deoxyribose or ribose, okay? So only one of 2 things there. So this is a basic monomer. This is a basic building block of a nucleic acid. So I know when we talk about DNA and RNA, we typically just refer to like the letter, like T, A, C, G, or U. But keep in mind that it also has these other 2 parts connected to it, okay? So the polymer can be DNA or RNA, and again, here's our nucleic acid there, our nucleotide. So it has the phosphate, the sugar, and then the nitrogen group. And those are going to be linked together, phosphate to sugar. So that kind of forms a backbone, right? That forms a backbone. And then the nitrogen groups form what we like to call the rungs of the ladder, right? Like that. So a lot of the time, we're going to have a double helix, especially when we're talking about DNA. And then in RNA, we only have a single strand. It might form into a helix like this. It might fold on itself. RNA is not that stable, so it will break down over time. DNA, however, is more stable. So here's just a compare and contrast of DNA and RNA. So DNA and RNA both carry genetic information, but RNA is directly involved in protein synthesis. DNA only stays in the nucleus when we're talking about eukaryotic cells. It's only going to stay in the nucleus. The RNA can enter and leave the nucleus. DNA is a double helix, like her hair, and then RNA can be a single helix or single-stranded. Deoxyribose is in DNA, ribose is in RNA. DNA is made up of these 4 bases, and then RNA is made up of these 4 bases. So RNA is going to have uracil instead of thymine. And that's only because thymine is a little bit bigger and uracil is smaller and cheaper to make in molecular terms. So that's really the only difference. Okay. So DNA is involved in what's called the central dogma of life. And the central dogma is basically saying that we go from DNA to RNA to protein. So that is the streamline of information, right? So we have DNA, we use that DNA to make RNA, we use that RNA to make protein. Okay, so that's what the central dogma is. Francis Crick first articulated the central dogma in the 50s. All right, so here's another nice video that goes over nucleic acids. All right, final poll question. A nucleotide of DNA may contain what? So a nucleotide, remember, is the monomer. DNA is the polymer. So each nucleotide contains what 3 things? And it has to be for DNA. So it's going to contain C. So deoxyribose, which is our sugar. That is a DNA base, thymine. And then phosphate group, of course. Okay, so hopefully now you should be able to describe nucleic acids, their structure, and the 2 different types, so DNA or RNA. Hopefully you can explain DNA's structure and role, and then RNA's structure and role. And then here is a nice GIF, which goes over the nucleotide structure, which again is your monomer. Okay, and that is it. So you can go on back through the lecture if you'd like. You can hit those review videos if you are unsure of some parts of the video. There are review games that I've created that you can use to review this, but make sure you complete your reading assignment, your lab, and your quiz for the week. Have a great day.